Hey everyone, my name is Hailey Pitre, and I did my independent research project on investigating the antimicrobial and antioxidant effects of pesticide-contaminated honey. So, honey is a sweet, viscous liquid made by bees from nectar, which undergoes a process involving enzymatic breakdown and dehydration. Honey's composition is mainly carbohydrates, water, proteins, small amounts of vitamins, and phenols. Honey is well known for its antimicrobial and antioxidant effects. Pesticides are a substance or mixture of substances intended for preventing, destroying, or controlling pests, causing harm during or otherwise interfering with production. Honey gets contaminated when bees travel for food and touch plants, crops, and flowers that have been sprayed with pesticides. Honey also can get contaminated when beekeepers use pesticides to control bee pests and diseases. And in some cases, these pesticides can cause resistance and make treatments less effective. Currently, there is a lack of studies investigating the effects that pesticides have on the antimicrobial and antioxidant effects of honey. The aim of this study is to evaluate the antimicrobial and antioxidant effects of pesticide-contaminated honey. It is expected that in the presence of pesticides and synthetic chemicals, the antimicrobial and antioxidant properties of honey will decrease. So to test this, I started by creating a pesticide spike sample. I tested alongside this an organic honey and a non-organic honey. So to start, I started by extracting the pesticides in the honey samples using Petrus extractions and running them on the GC to identify any pesticides. To test for antimicrobial properties, I started by doing an MIC assay on all the honey types, and I did this assay on a gram-negative bacteria and a gram-positive bacteria. I used E. coli and S. aureus. So after the MIC, I did a disc diffusion, which I followed that by a bacterial susceptibility test. And then to test the antioxidant effects, I did an FNC assay. So here in figure 1, I have the chromatographs of extracted pesticides. This is a representative image of the catcher's extraction. So in figure 1A, I have a negative control, B is the pesticide spiked, and image C is the non-organic honey, and D is the organic honey. There was no pesticides that could be identified, and there was no peaks observed in the organic or non-organic sample. There was also no results for the disc diffusion. So for the bacterial susceptibility test, for S. aureus, there was no difference before and after treatment. It had no effect on killing any of the S. aureus, but it did for E. coli. So after 24 hours, all the honey types significantly decreased the amount of colony-forming units. For the FNC, the non-organic honey had the highest amount of gallic acid equivalent, and it was significantly more than the organic or pesticide samples, which this could be assumed that this is because of the difference in floral origin, which may influence the phenolic synthesis. So, to conclude, the results of this study indicate that all honey types displayed antimicrobial activity against E. coli, and that the non-organic honey sample exhibited the highest antioxidant activity compared to both organic and pesticide spike samples. These results highlight the complexity of honey's bioactive properties and suggest that factors beyond organic certification, like floral source processing methods and environmental conditions, play a more significant role in determining its therapeutic potential. Overall, more testing should be done to more accurately determine the effects of contamination in honey. For some future directions, to more accurately determine the susceptibility of bacteria to honey, a time kill assay could be done with multiple time points to determine the amount of bacteria and how quickly it's being killed. And this may give us a better insight of the efficiency of the honeys. And another future direction is to identify the phenols present in the honey samples and analyze the sources and processing methods of the honey.